Now please write about isotonic solution. What is isotonic solution? Like if the medium which is surrounding the cell is having same concentration of water as the cell, so there will be no net movement of the water. So we will call such solution as isotonic solution. This solution is called isotonic solution. So there will be no net movement of water. Whatever is the water outside the cell, same is present inside the cell. So there will be no net movement. So cell size will remain the same. If I ask you, is there any change in the size of the cell? So there will be no change in the cell size. If we put in isotonic solution, you can just write this point by marking it a star.
Ma'am, what is that, ma'am? There will be no net movement of water.
Across. So if we put cell in isotonic solution, there will be no change in size of cell. So third type of solution is called hypertonic solution. So what is the definition of hypertonic solution? If the medium surrounding the cell has lower water concentration than the cell. Okay, this solution is called hypertonic solution. So what happened in this case? In hypertonic solution, the cell will shrink or will become small in size. The cell will shrink as water leaves the cell. So that state of shrinking of cell is called plasmolysis in plant cell, and it is called crenation in animal cell. So I am writing that also, that is very important. So what is plasmolysis? Plasmolysis plasmolysis. So the shrinkage,
Please show the previous slide.
yes.
Yes, ma'am. Now you can move on.
Okay, so plasmolysis is basically shrinkage of cell on placing, shrinkage of, you should write plant cell, shrinkage of water from the plant cell, और लिखना चाहते हो डिटेल में, I am just erasing it up. Shrinkage of plant cell on placing it in hypertonic solution. So I'm just writing for your for keeping it in your mind. See, plasmolysis, it starts from PL, and it gives you an indication that it happens in plant cell. Plasmolysis means shrinkage of the content. So whatever is the size of plant cell, it gets reduced when we put it in the hypertonic solution. So that is called plasmolysis. If I talk about crenation, so crenation is shrinkage of animal cell like RBC. Shrinkage of animal cell on placing it in hypertonic solution. So when we put it in hypertonic solution, then animal cell will shrink. So you can keep an example of RBC cell in your mind. So that is called crenation. Okay, so now let's come to the next definition, that is endocytosis. So what is endocytosis? It is the ingestion of material.
Amoeba.
Yeah, it is ingestion of materials by cell through plasma membrane. So it happens in amoeba, you know.
Yes, ma'am.
So now let's write about cell wall. So cell wall is like present only in plant cell. Okay, so cell wall is the outermost covering. Cell wall is non-living. And it is not selectively permeable, it is freely permeable. Plasma membrane is selectively permeable, but it is freely permeable. Cell wall is made up of cellulose. So it gives shape to the plant cell. So let me write the function of cell wall. So it makes the plant cell turgid. Provides mechanical strength. To the cell. Protection. So these are the basic function of the cell wall. So let's come to the nucleus. Nucleus is important because it has, you can call it as control center of the cell. So its shape is like spherical. It is a double membrane bond structure. It has two nuclear membrane. Okay, fluid which is filled inside nucleus. What is the name of that fluid? Fluid filled inside nucleus is called nucleoplasm, like cytoplasm, but that is filled inside nucleus is called nucleoplasm. Okay, so can we move?
Yes, ma'am.
So let's talk about chromosomes. वो वाला जो क्वेश्चन है जो अधिकतर पूछा जाता है, chromosomes. These are thread like structures. अगर क्रोमोसोम की डेफिनेशन आती है तो क्या लिखोगे? These are thread like structures present in the nucleus that becomes visible only during cell division. So once the cell get divided, so you can see chromosomes. So you can say they contain the hereditary information in the form of genes. Ayan, please read all your notes which are provided by the school properly. Don't leave anything unturned.
Okay, ma'am.
Ma'am, done.
Chromosome is made up of two components. DNA and proteins. So there are two things which make each chromosome, that is DNA and protein. ओके अब मैं एक डायग्राम बना देती हूँ क्रोमोसोम दिखता कैसा है दिस इज क्रोमोसोम तो ये इसकी दो आर्म्स हैं इन्हें क्या कहते हैं जो अप एंड डाउन है यू कैन सी दिस आर कॉल्ड क्रोमोसोम आर्म और जो बीच में है दैट इज कॉल्ड सेंट्रोमियर तो जब सेल डिवाइड होने वाला होता है तो ये डुप्लीकेट बना लेता है अपने आप को So is it done?
Yes, ma'am.
So जब सेल डिवाइड होने वाला होता है तो ये डुप्लीकेट बना लेता है इसका क्या मतलब है दो दो बना लेता है इट इज लाइक दैट तो इन्हें कहते हैं हम सिस्टर क्रोमेटाइड सो सी द क्रोमोसोम हैज डुप्लीकेटेड इटसेल्फ तो इसे हम कहेंगे डुप्लीकेटेड क्रोमोसोम ओके सो इट गेट्स कन्वर्टेड इनटू सिस्टर क्रोमेटाइड फिर जब सेल डिवीजन हो जाता है तो फिर ये अलग अलग हो जाते हैं सो वी कॉल डॉटर डॉटर मतलब जब वो इंडिपेंडेंट हो जाता है सो वी कॉल इट डॉटर आई एम जस्ट मेकिंग इट इंडिपेंडेंट सी नाउ यू ऑब्टेन टू क्रोमोसोम्स so we call it daughter chromosome so ये स्टोरी है क्रोमोसोम की एंड सी आई एम गिविंग यू आई जस्ट वांट टू